A research team led by Dr. Xiaoyu LI from the Research Division for Chemistry, Faculty of Science, in collaboration with Professor Yizhou LI from School of Pharmaceutical Sciences, Chongqing University and Professor Yan Cao from School of Pharmacy, 2nd Military Medical University in Shanghai has. The method developed by Dr. Li's team provides an efficient way to discover novel ligands and inhibitors against membrane proteins, which remain largely intractable to traditional approaches. The development of the methodology and its applications are now published in Nature Chemistry. Background membrane proteins on the cell surface perform a myriad of biological functions that are vital to the survival of cells and organisms. Not surprisingly, numerous human diseases are associated with aberrant membrane protein functions. Indeed, membrane proteins account for over 60% of the targets of all FDA-approved small molecule drugs. However, despite the significance, drug discovery against membrane proteins is notoriously challenging, mainly due to the special property of their natural habitat. In fact, membrane proteins have long been considered as a type of undruggable targets in the pharmaceutical industry. In recent years, DNA-encoded chemical library. To simplify, we can use a book library as an example. In a library, each book is indexed with a catalog number and spatially encoded with a specific location on a bookshelf. Analogously, in a DEL, each chemical compound is attached with a unique DNA tag, which serves as the catalog number recording the structural information of the compound. Today, DEL has been widely adopted by nearly all major pharmaceutical industry worldwide. However, DEL also had encountered significant difficulties in interrogate membrane proteins on live. Two key findings, tracking and boosting. There are two hurdles that the team has overcome to enable the application of DEL on live. First, cell surface is not a smooth convex shape like a balloon. It is extremely complex with hundreds of different biomolecules with a rugged topology. Thus, locating the desired target on the cell's surface is like finding a. The team has overcome this target specificity problem by using a method they previously developed, DNA programmed affinity labeling, DPAL. This method utilizes a DNA-based probe system that can specifically deliver a DNA tag to the desired protein on live cells, and the DNA tag serves as a beacon to direct target-specific DEL screening. In other words, the team first installed a tracker on the target to achieve screening specificity. The second challenge is target abundance. Typically, membrane proteins exist in nanomolar to low micromolar concentration, which is far below the high micromolar concentration needed to capture the tiny fraction of binders among billions of non-binders in a library. To solve this problem, the team employed a novel strategy by using complementary sequences in the DNA tag on the target protein and the actual library, so that the library can hybridize close to the target, thereby boosting the effective concentration of the target protein. In other words, the tracker can not only help the library locate the target, but also create an attractive force to concentrate the library around the target, not being distracted by the non-binding population. In the publication, the team reports their detailed methodology development, and they also demonstrate the generality and performance of this method by screening a 42 million compound library against folate receptor, FR, carbonic anhydrase 12, CA12, and epidermal growth factor receptor, EGF. This approach is expected to broadly applicable to many membrane proteins. For example, classical drug targets, such as GPCRs and ion channels, may be revisited in a live cell setting to identify new drug discovery opportunities by harnessing the power of DEL. We expect the utility of this method is not limited to drug discovery, but also in academic research to explore challenging biological systems, such as oligomeric membrane protein complexes and cell-cell communications, said Dr. Xiaoyu LI. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.